Hello, my name is Lauren Spahn, and this is my project on the optimization of lignin precipitation with functional group control for use in bio-based polyurethane foams. As urbanization continues to grow across the globe, more cost-competitive and environmentally friendly alternatives to synthetic petroleum-derived polymers and ways to manufacture them are on the rise. One example is polyurethane foams, which are used across multiple industries such as packaging, automotive, and furniture. Lignin is an organic polymer that has the potential to replace petroleum-derived polymers in a variety of applications and can act as the polyol in the reaction with an isocyanate to form polyurethane. One way to increase the functionality of the lignin polyol is to increase the amount of hydroxyl groups in the molecule, which will increase the possible sites for cross-linking and therefore form more dense cross-link networks for rigid foam applications. Increasing the amount of hydroxyl groups can be accomplished through demethylation. Through this process, the CH3 is removed from a methoxy group. Then the remaining oxygen is protonated, resulting in an OH group. Lignin is readily available from the pulp and paper industry in the form of black liquor, which is highly alkaline and has a pH of about 13. In my experiment, I plan to use dodecaine thiol as a demethylation catalyst due to its activity under alkaline conditions. Demethylation at the beginning of precipitation, rather than later in the process, bypasses the need for multiple pH changes, therefore saving time and resources. The proposed reaction mechanism begins when the dodecaine thiol is deprotonated under alkali conditions. Then the nucleophile attacks the methoxy side chain of the syringel unit and the methyl group is removed via a nucleophilic substitution. Finally, the oxygen is protonated during the final acidification step using H2SO4. Lignin can be recovered from black liquor using a modified Ligno-Boost procedure, which reduces the black liquor pH and causes the lignin to precipitate. In the first step, 5 weight % dodecaine thiol and water were added to black liquor at 75 degrees Celsius and held for either 15 or 30 minutes to demethylate the lignin. Then the liquid was sparged with CO2 for 1 hour at 80 kPa to drop the pH to about 9. Afterwards, the temperature was raised to 85 degrees Celsius. H2SO4 and water were added to bring the pH to 1.7, and the mixture was reacted for an hour and a half. Finally, the precipitate was washed and filtered to recover the solid lignin product. Preliminary FTIR analysis was completed on all lignin samples in order to see the extent of demethylation. In the control sample, we can see the broad OH peak, aliphatic CH3, and phenolic OH, all characteristic of monolignals. The methyl group at a wavelength of about 1050 is characteristic of syringel. We can also see a CO ring of syringel at about 1330 and the CO ring of guaiacyl at about 1265. In the demethylated sample, we can still see the same characteristic functional groups as in the control sample, except for the aliphatic carbon peaks at about 27 to 2900. These peaks remain after washing the sample with either dilute sulfuric acid, ethanol, or hexane, indicating that the thiol group is likely still attached to the lignin molecule. We also cannot see a significant increase in the OH peak of the demethylated sample, which could point toward unsuccessful demethylation. The hydroxyl groups in the lignin will be quantified using P-NMR in order to be completely sure. In terms of the lignin precipitation results, the solid mass yield recovered during the demethylation experiments is relatively similar to the solid mass yield recovered during the control experiments. Therefore, we can determine that neither the thiol residence time nor the addition of the thiol itself has a significant effect on the total mass yield of the precipitation. In conclusion, this experimental procedure is repeatable and reliable and can be used as a basis for further experimentation with lignin precipitation. Additionally, there is no evidence yet that demethylation has been successful in increasing the amount of hydroxyl groups using this method. Aliphatic carbon peaks in the FTIR suggest that the thiol is still bonded covalently to the lignin molecule, signifying that the proposed reaction mechanism may be incorrect and needs to be revised. The next steps in this project involve analyzing the samples using Phosphorus Nmr. The lignin samples undergo a phosphorylation using the agent TMDP so that the OH groups can be easily viewed as phosphate groups. The resulting peaks will be identified relative to a known internal standard and integrated in order to determine a quantitative value for OH content. If the demethylation is determined to be successful, then we will proceed with forming the lignin polyurethane foams. The lignin polyurethane foams will be prepared in various ratios of synthetic polyol to biopolyol, in order to determine how much lignin can be added while still maintaining the mechanical properties of synthetic polyurethane foam. Thank you for listening to my presentation.